that brings us back to what I want to talk to you about today. I want to talk to you about the gene alterations in lung cancer, how the gene alterations in our immune system dance together essentially to keep us healthy or let sometimes the cancers grow, and how we exploit those alterations for therapy, the gene-based alterations for therapies, and studying the immune alterations, see how we can make the immune system be more active in killing cancer cells. So let's go back to the basics. Genes are the basic molecules present in each and every cell in our body. And these genes that are in the human, what we call genome, the entire repertoire of genes, we have 20,000 genes. And these genes, roughly speaking, 20,000 genes, these genes produce what are called proteins. If you remember your biology class, the genes are coded DNA molecules, and they in turn make the RNA molecules, and they in turn make the proteins. Proteins are the business end of the whole thing, and proteins really make things do what the cells need to do. So of those 20,000 genes that are present in each and every cell in our body, these 20,000 genes, they do many things. They keep the cells active, they make the cells do what they're supposed to do, and then a time comes, the cells quietly die away, and then the new batch comes in. That's how our body works. Cells, by the way, are the basic building blocks in our body. Cells are like a bricks. If you're a huge building, cells are bricks. And these cells constantly undergo renewal, what we call renewal, which means the old cells die and the new cells come on. And there are mother cells that produce the daughter cells, and by copying the genetic material or the DNA. And when they do that, the mother cells or stem cells make the daughter cells. They have to copy all the 20,000 genes. When they do that, they sometimes make mistakes. But there are very good systems within the genes themselves that clean up all the mistakes. And the cell pretty much gets it right. The daughter cell gets it pretty much right, and 99.999%, everything is perfectly copied. But now and then there are some mistakes that creep in. What we call the DNA repair genes don't repair them, and then they eventually accumulate over time, and that's what leads to eventually, messed up proteins. Remember, I told you DNA makes RNA, RNA makes proteins, so bad DNA makes bad proteins, and those proteins get messed up, and they do things that they're not supposed to do. So that's what leads to diseases, not just cancer, or other diseases too. And cancer is a great example of how the genes get messed up and how we can really make out this distinctive differences between a normal gene and the abnormal gene by studying a normal tissue from a patient and the cancer tissue, and we can study this. So the DNA is coded basically in 4 letters: A, T, C, G. And A pairs with T and C pairs with G. So there are some rules and they follow. And now and then things get messed up. For example, in this stretch of the DNA or the gene, a stretch of DNA is a gene. And let's say, imagine this is a gene. And this stretch, there's supposed to be C, but then because of some mistake, instead of C, G comes on. And that results in alteration in the protein, and then that messes up the cells. And this is called a mutation. Mutation means a change. And here is a change in one of those letters that we see. In some instances, by the way, let me go back to that. Let's say, for example, you have EGFR. You may have heard of this L858R mutation. It's a kind of a mutation like that, where a single letter gets changed, and that leads to a different kind of uh, uh, what we call amino acid that makes proteins. So essentially, when the genes get messed up, it causes messed up proteins, and that's what happens, what we call a point mutation, a single alteration here. Now and then, what could happen is the gene can get amplified. Normally, for every gene, we have 2 copies, one from the mother, one from the father. But so every cell will have 2 copies of the gene. Sometimes the gene can get amplified. That's what you hear in breast cancer, HER2 gene amplified. So there are more than 2 copies, like 5 copies, 10 copies, in some instances, 100 copies. We won't go into the details of why that happens. There is a complex process that we have really now understood how these things happen, and that leads to too much of these genes that overactive and make the cells grow more and more. It's like giving too much of gas, in the car, and then the cells keep growing faster and faster because this particular gene is amplified. And there are some genes in our body that protects these cells from growing out of control. We call them, very easy to understand, tumor suppressor genes. And these are genes that are well known to suppress the cancer being formed. And sometimes through these processes, that good genes get destroyed or deleted. And that happens a lot in a gene called P53, a gene that is dubbed very famously as a guardian of the genome. In other words, this is the ultimate sentry, ultimate guard, and protecting the cell from all kinds of bad things. And that gene can be deleted, and that's called, one of the genes is P53, and that can also make the cells be more prone to develop cancer. And then there are other instances where a particular part of this gene stretch, we call long stretches of DNA, or we'll call this kind of a stretch, long stretches would make a gene, and long stretches of genes make a chromosome. And sometimes part of this chromosome can get altered. Either a piece can go from here to here, or the piece can revert itself, and that can make the genes more active or the proteins more active. And that's an instance that happens with the ALK gene, where it gets rearranged a little bit and that make the gene more active. And I won't go too much into the details in interest of time. There are other ways genes can get activated by what we call epigenetic alterations. If you think about it, your skin cells have the same 20,000 genes as your brain cells. But if you think about the skin, what the skin does is vastly different than what your brain does. How does it happen? Because in the brain, the genes that are not important for the brain function are silenced. In the skin cells don't have to think, don't have to beat like the heart cells beat. So they silence a lot of those genes, and they're just there to cover your body and protect it from any other external damages. So that those genes are the ones active. So out of the 20,000 genes, not all of them are active in every healthy cell in our body. Some are more active than the others, and that is done through this, what we call epigenetic alterations, where certain genes are silenced, certain genes are more active. What happens in cancer is that some genes are inappropriately silenced, some genes are inappropriately not silenced. What should be quiet is amplified, what should be active is deleted. And that is called epigenetic alterations. That happens in many cancers, and we won't get into that. But mostly, a lot of the genes get altered because there is a mutation where a single letter code gets messed up, or they have amplification or some deletion, or some genes get rearranged so that some proteins get more active. So that's basically what leads to cancer. So you can ask, why does this happen? No, I never smoked. I never drank. I ate. I'm a vegan. I did exercise. Why did I get lung cancer? There are a couple of ways you can explain this. One, we probably don't know what other factors in the environment leads to cancer. For example, radon exposure in high levels can cause cancer. Exposure to heavy metals can cause cancer, but they are not the majority of that. But a lot of these cancers happen randomly because, remember I told you, a mother cell has to break, and then a mother cell has to divide and produce daughter cells. And during that process, the mother cell has to copy 20,000 genes, and they make mistakes. And by the way, they do it within a couple of days. And when they make all the 20,000 gene copies, that's about 6 billion what we call base pairs. And when they do that, they make random mistakes, and they're not cleaned up, and they accumulate over time. And so it is a bit of a random process. To some extent, even if we eliminate all bad things on this planet Earth, we still have some cancer left. Substantially, the cancer burden will come down, no question about it. But still, there'll be some baseline cancers because of the inherent nature of the cell division, growth, and this proneness to get mistakes. And that happens to some extent.